The ocean covers more than 70% of our planet, yet most of it exists in darkness. Far below the surface, there are layers where oxygen dissolves into seawater in such small amounts that most animals would suffocate within minutes. These are known as oxygen minimum zones, vast stretches of mid-depth water where breathing becomes almost impossible. In certain parts of the Pacific, measurements fall to less than 1% of what we breathe at sea level. Research vessels have lowered instruments into these regions and watched oxygen readings nearly flatten. These zones are not small. Some extend across thousands of miles, shaped by ocean currents, temperature layers, and the slow sinking of organic matter from above. For decades, scientists believed these depths were nearly lifeless. Nets returned almost empty. Sonar detected little movement. The water seemed suspended between presence and absence. Then cameras began to show something unexpected. Movement, faint glows, shapes drifting where almost nothing should survive. Some creatures here slow their metabolism to conserve every trace of oxygen. Some carry blood pigments that bind it with unusual efficiency. Some barely move at all. As the planet warms, warm water holds less oxygen than cold, and over the past 50 years, parts of the ocean have measurably lost breath. A vampire squid drifts within these zones, gathering falling debris with delicate filaments instead of hunting. A barreleye fish hovers nearly motionless beneath its transparent dome. A vast ribbon-like jelly moves with minimal demand, surviving where others cannot enter. But if oxygen nearly disappears, why does life not disappear with it? What allows bodies to function where readings approach zero? How do entire ecosystems persist inside water once thought empty? And if the ocean continues losing oxygen as temperatures rise, what will remain? Tonight, we are traveling into the ocean's expanding oxygen minimum zones, a hidden layer where survival narrows to the smallest margin. But before we begin, if this space brings you peace, a like or subscribe helps other curious dreamers find their way here too. And I'd love to know, where are you listening from tonight? What time is it where you are? It's always lovely to see how far this calm corner travels. So now take a deep breath, let your shoulders loosen, and together let us drift gently into the ocean's fading breath. Before any creature drifts into view, the story rests on something invisible. Every breath taken at the surface leaves a faint imprint on the sea. Oxygen does not belong only to air, it dissolves into water, slipping between molecules, spreading through the upper layers in delicate balance. Waves fold it downward, wind presses it in. The boundary between sky and ocean becomes a place of quiet exchange where gases pass back and forth without sound. Marine animals rely on this dissolved oxygen as surely as lungs depend on air. Fish draw water across their gills, where thin membranes allow oxygen to enter the bloodstream. Invertebrates absorb it through feathery structures or even directly across their skin. Within cells, oxygen feeds the slow combustion that releases energy from food. Without it, muscles falter and nerves dim. Life in water is bound to the same chemical necessity that governs life on land. Yet the ocean does not breathe evenly from top to bottom. Sunlight warms the surface, creating a lighter layer that floats above colder, denser water. This separation forms a boundary known as the thermocline, a gradient that resists mixing. Oxygen added near the top does not easily travel downward. The deeper layers may remain isolated for years, even decades, depending on currents and large-scale circulation. At the sunlit surface, microscopic plants called phytoplankton perform another exchange. Through photosynthesis, they use light to transform carbon dioxide into organic matter, releasing oxygen as a byproduct. For a time, the upper ocean becomes gently enriched. But when these tiny plants die or are consumed, fragments sink. A steady rain of organic particles drifts toward darker waters below. As this material descends, bacteria begin their work. They decompose the sinking matter, drawing on dissolved oxygen to fuel the process. In regions where productivity at the surface is high and mixing is weak, consumption outpaces replenishment. Oxygen levels fall, sometimes to a thin trace. Such areas are known as oxygen minimum zones. They arise naturally in parts of the world where currents and climate align in particular ways. Along the eastern margins of the Pacific Ocean, persistent winds drive nutrient-rich water upward, stimulating surface growth and, in turn, heavy sinking flux. In the Arabian Sea, seasonal monsoon patterns and restricted circulation create similar conditions. Mid-depth waters, often between several hundred and 1,000 meters, can hold the lowest concentrations, caught between limited ventilation from above and slow renewal from below. These natural zones differ from coastal dead zones driven by pollution. Near river mouths, excess nutrients from agriculture and industry trigger intense blooms that rapidly collapse, stripping oxygen from shallow seas. The mechanisms share a pattern of imbalance, though their origins are distinct. Within this layered architecture of supply and demand, the contours of a breathless realm take shape, quiet and precise, waiting to be understood. There is a small figure suspended in the dim layers where oxygen thins to a trace, neither rushing nor retreating, simply remaining. Its cloak of dark velvet stretches between slender arms, forming a soft web that gathers the faintest currents. Pale blue eyes reflect what little light filters down, steady and unhurried. This is Vampiroteuthis infernalis, a creature whose name suggests ferocity, yet whose existence is defined by restraint. It inhabits the heart of oxygen minimum zones, not at their edges, but within their quiet core. While many animals skirt these waters, it resides there continuously, adapted to conditions that would silence faster lives. Its body is gelatinous and light, reducing the cost of movement. Instead of powerful jets or swift pursuits, it drifts with patient control, adjusting position through gentle pulses. The metabolism within is remarkably slow. Oxygen is consumed sparingly, each molecule used with measured care. In laboratory observations, individuals have endured concentrations that approach the limits of animal tolerance, remaining composed where others would falter. Inside its veins flows hemocyanin, a copper-based pigment that binds oxygen efficiently even when concentrations are scarce. The chemistry allows uptake at levels that would leave many fish gasping. Large gill surfaces and a high affinity for oxygen ensure that even faint traces can be gathered and carried to waiting tissues. The rhythm of its life proceeds at a pace tuned to scarcity. Feeding follows the same principle of conservation. Rather than chasing prey, the vampire squid extends two delicate filaments from hidden pockets within its arms. These threads, fine and sticky, unfurl into the surrounding water like drifting silk. They collect particles of marine snow, fragments of organic matter descending from above. When the filaments are drawn back, the gathered debris is wrapped into small bundles and guided to the mouth. Nothing is hurried. Energy is neither wasted nor sought in excess. If disturbed, it does not flee with explosive speed. Instead, it may invert its webbed arms over its body, presenting a posture once likened to a dark flower turned inward. Bioluminescent organs along the arm tips can release brief points of blue light, subtle distractions in a place where brightness is rare. Even this display is economical, flashes measured and sparse. In the stillness of low oxygen water, this quiet cephalopod persists without urgency. It does not conquer its environment, it aligns with it. Through efficiency, chemical precision, and deliberate motion, It demonstrates that survival here is not an accident, but a refined balance. Within waters long assumed to be nearly empty, a small velvet figure endures, steady as the slow fall of unseen snow. Light bends strangely around the next inhabitant, as though water itself has become glass. Within the dim midwater, a small fish holds position with delicate precision. Its body is dark and modest, scarcely longer than a human hand, yet the front of its head forms a clear, rounded shield. Through this transparent dome, internal structures are visible, pale, and luminous in the surrounding gloom. This is the barreleye fish, often identified as Macropinna microstoma, a creature shaped by scarcity and shadow. Inside the dome rest two tubular eyes, bright green and cylindrical, pointing upward like twin periscopes. They are not set on the sides of the head, as in most fish. Instead, they gaze toward the faint glow filtering from above, scanning for silhouettes that drift against the distant brightness. The transparency of the shield protects these sensitive organs from stinging tentacles and drifting debris, while allowing light to pass inward with minimal obstruction. The fish does not chase in wide arcs or dart through open water. It hovers, almost motionless, stabilizing itself with small adjustments of its fins. Each movement is measured. In layers where oxygen declines, economy becomes instinct. Muscle mass is modest, its form compact, reducing the demand for constant exertion. By remaining suspended rather than actively swimming, it conserves what little oxygen is available. When prey approaches, often small crustaceans or fragments entangled in siphonophore tentacles, the eyes can rotate forward within the dome. This subtle shift changes their orientation from upward scanning to forward focus, allowing precise feeding without the need for rapid pursuit. The motion is internal, quiet, more pivot than lunge. The dome itself is filled with clear fluid, cushioning the eyes and maintaining optical clarity under pressure. Its surface appears fragile, yet it endures the weight of hundreds of meters of water. Evolution has shaped it, not for speed or armor, but for efficiency and vision in constrained conditions. Observations from remotely operated vehicles reveal individuals lingering near the boundaries of oxygen-poor layers, neither at the surface nor in the deepest reaches. There, food drifts in unpredictable paths, and competition from faster predators lessens. In this narrow band, patience holds value. Seen through the lens of a submersible camera, the barreleye seems almost unreal, a small lantern enclosed in crystal. Yet its existence is grounded in physiology and physics, in the chemistry of breath and the geometry of light. It survives not by overpowering its environment, but by aligning its senses and energy use with the thinness of the water around it. Within a realm once assumed to be spare and featureless, a clear-headed watcher remains suspended, steady as the slow turning of its inward eyes. Sometimes the camera reveals not a body, but a movement that seems to rearrange the water itself. Out of the dim expanse emerges a vast, translucent form, pale as moonlit silk. It unfolds gradually, as though distance and scale are difficult to measure in this suspended world. This is the giant phantom jelly, known to science as Stygiomedusa gigantea, a creature so rarely encountered that each sighting feels like an accidental meeting. Its bell is broad and softly rounded, drifting with unhurried pulses that barely disturb the surrounding sea. Extending from beneath are four immense, ribbon-like arms, each trailing many meters behind. They do not lash or coil. They stream outward in slow arcs, forming curtains that filter whatever small organisms wander into their reach. Unlike many surface-dwelling jellyfish, This species lacks the dense clusters of stinging tentacles that require active contraction. Its anatomy is simplified, composed largely of water bound within delicate tissues. The gelatinous structure demands little oxygen to maintain. There are no heavy muscles to sustain, no swift chases to fuel. Instead, its body functions as a living net, suspended in place by gentle buoyancy and minor adjustments of its bell. The phantom jelly does not pursue prey. It waits. Small crustaceans and drifting animals encounter the long oral arms and become entangled in mucus-lined folds. From there, the arms convey the captured food toward the central mouth with gradual contractions. Each action unfolds slowly, in keeping with the limited energy available in these midwater depths. Encounters have occurred in oxygen, poor regions where many active swimmers are scarce. In such layers, the advantages of stillness become clear. A creature built of soft tissue and minimal metabolic demand can persist where more muscular forms would struggle. The phantom jelly's respiration occurs across thin surfaces, oxygen diffusing directly into its tissues from the surrounding water. Even at reduced concentrations, diffusion can suffice for a body that asks so little. Under the lights of a submersible, its transparency becomes luminous. Internal canals branch, like faint rivers, through its bell, distributing nutrients in measured currents. The long arms sway independently, describing vast arcs that suggest size without aggression. It appears less like an animal in motion and more like a drifting architecture, assembled from water and intention. In this quiet stratum, survival is written in softness. The giant phantom jelly embodies a principle already glimpsed in smaller forms, that endurance here favors those who spend little and gather patiently. Its presence alters the perception of emptiness, revealing that even in thin water, life can take on grand dimensions without haste or force. In these layered waters, motion carries a cost that is counted molecule by molecule. Every contraction of muscle requires oxygen. Within cells, mitochondria draw it inward to unlock energy stored in food, releasing the force that bends a fin or snaps a jaw. When oxygen is plentiful, the transaction proceeds easily. In thin layers, each movement becomes an expenditure that cannot be quickly replaced. Fast swimming accelerates this demand. Muscular fish rely on dense fibers rich in myoglobin, beating tails in steady rhythm to generate thrust. The chemistry behind such power consumes oxygen at high rates. In regions where dissolved concentrations fall to a fraction of surface values, sustained speed would drain available reserves in moments. The body responds with rising carbon dioxide and shifting acidity, signals that limit endurance. For this reason, many inhabitants of oxygen-poor zones adopt a different strategy. They drift or hover, reducing the frequency and intensity of muscle contraction. Fins undulate gently, enough to maintain position, but not enough to ignite rapid respiration. The heart slows. Blood circulates with quiet efficiency. Energy use drops to a baseline that matches the faint supply available in surrounding water. Body composition reflects this economy. Gelatinous forms contain large volumes of water, supported by thin connective tissues, with minimal, dense muscle. Their buoyancy is nearly neutral, so remaining suspended demands little effort. Diffusion across broad surfaces can meet modest oxygen needs without elaborate pumping systems. In contrast, heavily muscled predators face structural requirements that bind them to higher metabolic rates. In low oxygen layers, such designs become liabilities. Over generations, natural selection favors those whose physiology aligns with scarcity. Individuals that conserve energy survive longer and reproduce more successfully. Metabolic pathways adjust, favoring aerobic processes that operate efficiently at low partial pressures of oxygen. Enzymes become tuned to function under conditions that would inhibit other species. Some animals reduce spontaneous activity to the lowest possible level, emerging from stillness only when opportunity passes directly within reach. Even buoyancy mechanisms reflect this pattern. Reduced bone density, high lipid content, and gas-filled structures allow suspension without continuous swimming. Neural systems emphasize sensitivity over speed, detecting faint vibrations rather than pursuing distant signals. Feeding strategies shift from pursuit to interception, from chase to capture by contact. In such an environment, stillness is not absence, but adaptation. Movement remains possible, yet it is measured and deliberate. The water does not forbid action, it imposes limits that shape it. Through restraint, organisms balance internal demand with external supply. The apparent quiet becomes a form of equilibrium, a choreography in which survival depends less on force and more on careful accounting of breath. Inside each drifting body, another landscape unfolds, one too small to see, yet decisive in its quiet work. Oxygen enters not only through gills or skin, but into blood, where specialized molecules wait to bind it. In many surface-dwelling fish, and in all mammals, that molecule is hemoglobin, an iron-based protein that turns red when carrying oxygen. Its structure is well suited to waters rich in dissolved gas, releasing oxygen readily to active tissues that demand constant supply. In the dim layers where concentrations fall, a different chemistry often prevails. Many invertebrates and some deep-sea animals rely on hemocyanin, a copper-based pigment that circulates in pale blue tones when oxygenated. Hemocyanin binds oxygen with high affinity at low partial pressures, meaning it can capture and hold molecules even when they are scarce. This property becomes essential in water, where each dissolved particle is precious. Affinity, however, must be balanced with release. If oxygen is held too tightly, tissues cannot access it. Species adapted to thin waters fine-tune this balance through subtle changes in protein structure and blood chemistry. Variations in pH, temperature, and ion concentration shift how readily oxygen is loaded and unloaded. The Bohr effect, a principle describing how acidity influences binding, operates here with particular sensitivity, ensuring delivery continues even as environmental conditions fluctuate. In some fish that frequent oxygen minimum zones, gill surfaces expand relative to body size. Filaments grow longer and lamellae become more numerous, increasing the area across which oxygen can diffuse. Blood vessels within these structures lie close to the water, minimizing the distance a molecule must travel. Circulatory systems adjust as well, with hearts capable of maintaining flow without excessive strain. At the cellular level, mitochondria may be arranged to optimize oxygen use, clustered near capillaries to shorten diffusion pathways. Enzymes involved in aerobic metabolism display efficiencies that allow energy extraction from limited supply. Some species reduce reliance on pathways that demand high oxygen flux, favoring those that operate steadily at lower rates. Red blood cell counts can vary, as can the concentration of respiratory pigments within plasma. These adjustments alter the overall oxygen carrying capacity of the bloodstream. Even membrane permeability shifts, allowing faster passage of gases into tissues. Each modification alone may seem modest, yet together they form an integrated system aligned with environmental constraint. In this way, blood becomes not merely a transport medium, but a refined instrument tuned to scarcity. Within veins and vessels, chemistry compensates for absence, translating faint external traces into usable energy. The body, from gill surface to mitochondrial matrix, reflects an intimate dialogue with its surroundings. Life persists here not through force, but through molecular precision, each adaptation woven into the quiet rhythm of thin water. There are moments in these depths when life does not speed up to endure, but instead grows quieter. Within certain fish that inhabit oxygen-poor layers, the heart itself changes tempo. Beats that might pulse steadily in well-ventilated waters become spaced and deliberate. This reduction in heart rate, known as bradycardia, lowers the volume of blood moving through the body each minute. Circulation slows, and with it the overall demand for oxygen. The adjustment is not a sign of weakness, but of control. Cardiac tissue responds to chemical signals carried in the bloodstream, sensing diminished oxygen tension and altered carbon dioxide levels. In response, neural pathways modulate the pacemaker cells that govern rhythm. The heart settles into a slower cadence that matches the limited availability of dissolved gas outside. Metabolic suppression accompanies this shift. Cells reduce their rate of energy consumption, turning down processes that are not immediately essential. Protein synthesis may decline. Growth pauses. Even spontaneous movement lessens. Mitochondria continue their work, yet at a pace aligned with scarcity. Instead of operating at maximum capacity, they function in a restrained mode, extracting energy gradually from stored substrates. This physiological state resembles a controlled dimming rather than a shutdown. Organs remain active, but at a baseline sufficient for maintenance rather than expansion. Glycogen reserves are conserved. Lipid stores are metabolized carefully. The balance between supply and demand is recalibrated so that intake, though minimal, remains adequate. Some species can tolerate brief intervals of even lower oxygen by temporarily engaging anaerobic pathways, producing energy without oxygen for short periods. The byproducts of such metabolism are managed through buffering systems in blood and tissues, preventing harmful accumulation. Once oxygen levels stabilize, aerobic processes resume their central role. Energy rationing becomes a way of life. Feeding events may be infrequent, yet the body stretches each gain across extended intervals. Digestive activity proceeds slowly, allowing nutrients to be absorbed with efficiency. Hormonal signals coordinate these adjustments, integrating cardiovascular, respiratory, and metabolic systems into a unified response. Observations from controlled studies reveal that when oxygen concentrations rise again, heart rates and metabolic rates can increase, demonstrating flexibility rather than permanent alteration. This capacity to modulate internal tempo offers resilience in environments where conditions fluctuate. In these subdued waters, survival depends not on constant exertion but on careful pacing. The slowed heartbeat echoes through vessels like a distant metronome, steady and economical. Through restraint at the level of rhythm and chemistry, certain creatures endure on the thinnest margins, sustaining life with a measured quiet that aligns precisely with their surroundings. As night gathers over the surface of the sea, a vast ascent begins far below, quiet yet immense beyond measure. Across the world's oceans, billions of small animals rise from depth toward the upper layers under cover of darkness. Lanternfish, bristlemouths, shrimp, squid, and countless gelatinous drifters participate in this daily vertical migration. Each individual is small, many scarcely longer than a finger, yet together they form the largest synchronized movement of biomass on Earth. By day they remain deeper, where dim light offers protection from visual predators. With sunset, they begin their climb. Fins beat steadily, bodies angle upward, and entire communities shift position through hundreds of meters of water. The migration unfolds every evening and reverses at dawn, a rhythm repeated without pause across seasons and hemispheres. For those that dwell near oxygen minimum zones, this journey requires passage through layers where dissolved oxygen thins. As they rise, they cross gradients in temperature and chemistry, encountering regions that demand physiological flexibility. Species adapted to these boundaries adjust heart rate, ventilation, and blood chemistry in transit, sustaining movement despite fluctuating supply. The migration is not uniform. Some species complete the full ascent to near surface waters, feeding on phytoplankton and smaller zooplankton under darkness. Others travel shorter distances, remaining within intermediate depths, yet still participating in the collective shift. The vertical span may range from a few dozen meters to nearly a kilometer, depending on region and season. Acoustic instruments lowered from research vessels detect this movement as a shifting layer of echoes, once mistaken for the seafloor itself. As animals rise, the scattering layer ascends. As they descend, it sinks. The pattern repeats daily, revealing the magnitude of the event through sound rather than sight. In oxygen-poor regions, the migration can compress into narrower bands where conditions remain tolerable. Animals cluster along boundaries that balance access to food with survivable chemistry. Predators follow these moving concentrations, and nutrients are transported upward and downward as organisms feed and excrete along their route. Carbon fixed near the surface becomes redistributed through depth, linking distant layers through motion. The scale of this exchange extends beyond individual lives. Each night, an immense volume of living tissue shifts position, carrying energy across gradients of light and oxygen. The movement is coordinated not by command, but by environmental cues, primarily the fading and return of sunlight. Even in waters where oxygen declines, the migration persists, adjusted yet continuous. Through this daily procession, the ocean reveals a hidden circulation of life layered within its physical currents. Billions rise and fall in measured cadence, threading through thin waters without spectacle, sustaining a planetary rhythm that unfolds in darkness and returns with dawn. Long before cameras reached these depths, sound offered the first hint that something vast was suspended in the dark. When early oceanographers sent pulses of sonar downward, the echoes returned from what appeared to be a solid boundary far above the true seabed. Instruments traced a continuous layer stretching across entire basins, rising toward the surface at night and sinking again with daylight. At first, it was mapped as bottom topography, a false floor where none existed. Only later did scientists realize the signal came from living bodies. This phenomenon became known as the deep scattering layer, a broad band of midwater biomass dense enough to reflect acoustic waves. The echoes are generated by swim bladders in fish, by the bodies of crustaceans, by gelatinous organisms whose tissues differ subtly from the surrounding water. Individually, they are small, yet together they form a layer thick with life, often hundreds of meters wide and extending across vast horizontal distances. Within regions of reduced oxygen, the structure of this layer changes shape. Instead of occupying a wide vertical span, animals may cluster along narrow chemical gradients where conditions remain tolerable. The boundary between slightly higher and lower oxygen becomes a corridor of concentration. There, organisms gather in remarkable density, compressed between scarcity below and predation above. The effect can be measured not only by sound, but by nets and cameras lowered carefully into position. Lanternfish, siphonophores, shrimp, squid, and delicate larvae share the same constrained space, suspended in proximity that would be unlikely in more uniform waters. Competition and cooperation both intensify in such bands, as food and safety align along the same thin horizon. The deep scattering layer participates in the daily vertical migration, ascending after dusk and descending before dawn. In oxygen minimum zones, however, its lower boundary may be set by physiological limits. Animals descend only as far as their blood chemistry and metabolic control permit. The result is a living contour, shaped by invisible gradients rather than by rock or sediment. Acoustic profiles reveal that this biological layer can rival the reflectivity of the seafloor itself. The density of organisms is sufficient to alter the path of sound, creating a dynamic landscape that shifts with time of day and season. Beneath what once seemed like emptiness lies an architecture built from muscle, chitin, and gelatin, arranged by chemistry. In these waters, where oxygen narrows the available space, life does not vanish, it reorganizes. The deep scattering layer stands as evidence of hidden abundance, a concentrated weave of bodies occupying a delicate balance. Through sound alone, the ocean disclosed that even in thin water, its interior is crowded with motion held in careful suspension. Not all respiration depends on oxygen. Within the dim chemistry of these waters, countless microbes carry out forms of metabolism that unfold without a single molecule of O2. They are anaerobic bacteria, organisms whose existence is tuned to environments where oxygen fades and other compounds take its place. Invisible to cameras and silent to sonar, they shape the character of oxygen minimum zones from within. In oxygen-rich seas, microbes commonly use oxygen as the final electron acceptor in cellular respiration, completing the chain of reactions that release energy from organic matter. In thin waters, that pathway narrows. Certain bacteria turn instead to nitrate, a dissolved form of nitrogen carried through the ocean from rivers, upwelling currents, and biological recycling. Through a process known as denitrification, they use nitrate in place of oxygen, stripping it of oxygen atoms to generate energy. The chemistry unfolds step by step. Nitrate is reduced to nitrite, then to nitric oxide, nitrous oxide, and finally to nitrogen gas. Each transformation is catalyzed by specialized enzymes operating within microbial cells. The final product, nitrogen gas, drifts upward and eventually escapes to the atmosphere, completing a cycle that links ocean and air. Another pathway, anemox, short for anaerobic ammonium oxidation, combines ammonium with nitrite to produce nitrogen gas directly. Discovered only in recent decades, this process revealed that entire microbial communities can thrive in zones once thought chemically stagnant. These organisms construct internal compartments where sensitive reactions proceed in isolation, protecting their enzymes from interference. Through such pathways, microbes regulate the availability of bioavailable nitrogen in the ocean. By converting fixed nitrogen compounds back into inert gas, they influence primary productivity at the surface, where phytoplankton depend on nitrate and ammonium to grow. Thus, activity within oxygen minimum zones reverberates upward, shaping ecosystems far above. These reactions also produce intermediates, such as nitrous oxide, a greenhouse gas, with implications for atmospheric chemistry. In regions where oxygen is low but not absent, gradients create niches for different microbial guilds, each occupying a narrow chemical band. The result is a stratified microbial landscape layered within the water column. Although invisible, the scale of this activity is immense. Trillions of cells perform these conversions continuously, balancing inputs from rivers and upwelling with losses to the atmosphere. In doing so, they sustain the broader nitrogen cycle, one of the central engines of marine productivity. Within waters where animals slow their hearts and conserve breath, microbes proceed without it entirely. Their metabolisms trace delicate chemical pathways through scarcity, transforming compounds molecule by molecule. In the quiet of oxygen minimum zones, life persists not only in drifting forms, but also in reactions too small to see, yet vast in consequence. In waters where oxygen thins, dominance shifts quietly. Predators built for speed rely on powerful muscles and rapid bursts of motion. Such exertion draws heavily on oxygen, fueling fibers that contract with force and precision. In environments where dissolved concentrations remain high, this strategy succeeds. Energy flows upward through complex food webs, sustaining hunters that chase, overtake, and consume. Within oxygen minimum zones, that balance alters. The energetic cost of swift pursuit rises as available oxygen falls. Fast swimming demands increased ventilation, accelerated heart rate, and rapid circulation. In thin water, these responses strain physiological limits. A hunter that expends too much too quickly may exhaust its supply before capture is secured. As a result, the structure of the food web becomes simplified. Layers dense with midwater life do exist, yet interactions are shaped by chemical constraint. Fewer large, highly active predators maintain residence in these zones. Those that enter often do so transiently, passing through rather than remaining. The permanent inhabitants tend to favor strategies that reduce continuous expenditure. Energy scarcity further shapes this pattern. Primary production that ultimately supports midwater communities begins at the surface, then descends in attenuated form. By the time organic matter reaches these depths, much has already been consumed or decomposed. The caloric value available to sustain large, metabolically demanding hunters is limited. In such conditions, selection favors efficiency over aggression. Predation does occur, yet it is often opportunistic rather than relentless. Sit and wait strategies replace long chases. Ambush becomes more common than pursuit. Some predators possess enlarged sensory organs capable of detecting subtle movements nearby, reducing the need for wide-ranging search. Others exploit vertical migration, feeding during brief intervals when prey densities increase along shared boundaries. The absence of constant high-speed predation influences prey behavior as well. Animals may cluster within narrow bands where oxygen remains tolerable, forming communities that balance access to food with reduced exposure to highly active hunters. The ecosystem settles into a dynamic equilibrium shaped less by rapid consumption and more by measured exchange. Even among species capable of stronger swimming, activity is often timed carefully. Short bursts replace sustained effort. Feeding bouts are followed by extended periods of stillness. Internal physiology mirrors this pattern, with metabolic rates adjusted to match intermittent demand. In this constrained environment, patience holds advantage. The chemistry of the water sets limits that ripple through trophic levels, shaping interactions from microbes to fish. Where oxygen restricts intensity, the dominance of swift predators diminishes, and the fabric of the ecosystem reorganizes around endurance and restraint. Sound travels farther than light in the ocean, threading through water in long, invisible arcs. In coastal shallows and coral reefs, the sea is rarely quiet. Snapping shrimp produce sharp cracks as they close specialized claws, generating cavitation bubbles that collapse with startling force. Fish call to one another with grunts, hums, and pulses created by vibrating muscles against their swim bladders. Together, these countless signals form a dense acoustic tapestry, a background alive with texture. Within oxygen minimum zones, that tapestry thins. As oxygen declines, species that rely on constant movement and active communication become less common. Snapping shrimp, which depend on oxygen-rich coastal habitats, are largely absent from midwater layers where dissolved concentrations fall to low levels. Reef fish and benthic invertebrates that generate persistent biological noise do not occupy these depths in significant numbers. The absence of their presence alters the character of the water. Hydrophones lowered into such regions record a different soundscape. Instead of continuous crackling and layered calls, there are longer intervals between distinct signals. Occasional movements, faint clicks, or distant low-frequency pulses pass through, yet the overall intensity diminishes. The acoustic environment reflects the metabolic restraint of its inhabitants. Even the vertical migration, immense in scale, produces limited sound compared to surface swarms. Many midwater organisms lack hard structures that resonate strongly. Gelatinous bodies absorb rather than reflect acoustic energy. Slow fin beats and measured pulses generate minimal turbulence. In thin water, silence becomes a byproduct of physiological economy. This quiet carries information. Marine mammals that traverse the ocean rely on sound to locate prey and navigate across vast distances. Changes in background noise may signal shifts in species composition or distribution. For scientists, long-term acoustic monitoring reveals patterns tied to oxygen variability, offering insight into how ecosystems respond to environmental change. Chemical conditions influence acoustics indirectly as well. Reduced oxygen can affect the buoyancy and distribution of organisms, altering where sound scattering layers form. As life compresses into narrower bands, acoustic reflections may intensify locally while fading elsewhere. The soundscape becomes uneven, shaped by gradients invisible to the eye. In these depths, silence is not emptiness. It is an ecological signature emerging from altered metabolism and simplified community structure. The water still carries currents, particles, and living forms, yet their collective presence registers more subtly in sound. Where reefs resound with crackling life, oxygen pore zones hold a muted resonance. The thinning of biological noise mirrors the thinning of dissolved gas, each connected to the other through the demands of respiration. In this restrained acoustic field, the ocean speaks softly, its quiet revealing the contours of life shaped by scarcity. Far above the quiet layers where oxygen thins, the ocean is in constant motion. Surface winds sweep across vast distances, pushing water into broad gyres that rotate slowly within each basin. Along certain coasts, persistent winds drive surface water away from land, drawing colder, nutrient-rich water upward from depth. These currents nourish life near the surface, yet they also set the stage for what unfolds below. Warm sunlight heats the upper ocean, creating a buoyant layer that rests atop denser, colder water. Between them forms a transition known as the thermocline, a region where temperature shifts rapidly with depth. This boundary resists mixing. Oxygen absorbed at the surface or produced through photosynthesis remains largely confined to the upper layer, while deeper waters become separated from direct atmospheric exchange. In some regions, the geometry of currents reinforces this stratification. Eastern boundary currents, flowing along the western edges of continents, are particularly associated with strong upwelling and high productivity. As organic matter sinks from these productive surface waters, microbes decompose it, consuming oxygen in the process. Because vertical mixing is limited, the consumed oxygen is not easily replaced. Deep waters do receive oxygen, but on far longer timescales. In polar regions, cold, dense water forms and sinks, initiating the global thermohaline circulation. This vast conveyor connects ocean basins, carrying oxygen-rich water into the deep sea. Yet the journey is slow. Once submerged, these waters may travel for centuries before returning to the surface. Along the way, oxygen is gradually used by organisms and microbes, and concentrations decline. Where circulation is sluggish and productivity is high, oxygen minimum zones can develop naturally. The eastern tropical Pacific and parts of the Arabian Sea exemplify this balance. In these regions, persistent stratification, strong nutrient supply, and limited deep ventilation combine to create midwater layers where oxygen reaches very low levels without any direct influence from pollution. Seasonal patterns also play a role. Monsoon-driven winds in the Arabian Sea alter circulation and productivity throughout the year, modulating oxygen concentrations at depth. Over longer climatic cycles, changes in temperature and circulation strength can expand or contract these zones, reflecting the ocean's sensitivity to subtle shifts in energy balance. Thus, oxygen minimum zones arise not from sudden events, but from the interplay of physics, chemistry, and biology unfolding over vast scales of time and space. Currents redistribute heat and nutrients, thermoclines limit exchange, sinking organic matter draws down oxygen, and deep circulation renews it only gradually. The result is a layered ocean whose breadth is uneven, shaped by forces that move quietly across centuries. West of the Indian subcontinent, where monsoon winds sweep across open water, a vast midwater layer holds remarkably little oxygen. The Arabian Sea contains one of the largest and most intense oxygen minimum zones on Earth. Beneath its productive surface, at depths often between 100 and 1,000 meters, dissolved oxygen concentrations can approach near zero. Instruments lowered from research vessels have recorded values so low that conventional sensors once struggled to measure them accurately. In these layers, the margin between aerobic life and chemical anoxia becomes exceedingly thin. The surface waters above are seasonally enriched by monsoon-driven upwelling. Nutrients rise from depth, fueling blooms of phytoplankton that spread across wide expanses. When this organic material sinks, microbial communities begin to decompose it, drawing down oxygen in the process. Strong stratification limits replenishment from above, and the renewal of deeper waters occurs only slowly through distant circulation pathways. Over time, the midwater becomes depleted. Oceanographic expeditions have traced the boundaries of this zone using conductivity, temperature, depth instruments paired with advanced oxygen sensors. Autonomous floats now drift through the basin, profiling oxygen concentrations across seasons and years. These measurements reveal a persistent core of hypoxia that expands and contracts subtly with climatic variability, yet remains a defining feature of the region. Within this chemically constrained environment, life persists in forms adapted to its limits. Zooplankton species exhibit high tolerance to low oxygen, adjusting their vertical distributions to remain within survivable bands. Certain fish reduce activity and alter blood chemistry, allowing residence where others cannot endure. Microbial communities flourish in the absence of oxygen, driving intense denitrification that removes fixed nitrogen from the water column at globally significant rates. The Arabian Sea oxygen minimum zone also influences fisheries along surrounding coasts. Species sensitive to low oxygen may shift their depth ranges or geographic distributions, compressing into narrower habitats. This spatial reorganization affects predator and prey interactions, as well as human livelihoods tied to marine resources. Sediment cores collected from the seafloor preserve records of past oxygen fluctuations, revealing that the intensity of this zone has varied over centuries in response to monsoon, strength, and broader climatic patterns. The present configuration reflects both enduring natural processes and contemporary environmental change. In this basin, chemistry and circulation converge to create a region where oxygen nearly vanishes, yet biological systems continue in adjusted form. The Arabian Sea stands as a vast natural laboratory, demonstrating how ocean physics, seasonal winds, and microbial metabolism combine to shape one of the planet's most pronounced low oxygen environments. Across the broad sweep of the eastern tropical Pacific, a quiet boundary stretches for thousands of kilometers beneath the surface. From the coasts of Central and South America westward into the open ocean, one of the planet's most extensive oxygen minimum zones occupies the midwater column. Its geographic reach spans an area comparable to continents, forming a continuous band where dissolved oxygen remains persistently low. The zone is not a sharp edge, but a layered expanse shaped by circulation and productivity that unfold over immense distances. Here, equatorial currents and coastal upwelling sustain high surface productivity. Nutrient-rich waters rise along the continental margin, supporting blooms that feed intricate food webs above. As organic matter descends, microbial respiration consumes oxygen at depth. The stratified water column limits vertical exchange, and the renewal of deeper layers proceeds slowly through basin-scale circulation. Measurements collected over decades reveal that hypoxia in this region is not fleeting. Long-term observational programs, including moored instruments and autonomous profiling floats, document consistently reduced oxygen concentrations between roughly 200 and 800 meters. Seasonal variability occurs, yet the core of the zone remains a stable feature of the basin. Deep submersibles have traversed these waters, offering direct glimpses into a realm defined by chemical constraint. Through viewports and camera systems, researchers observe midwater communities distributed along narrow gradients. Organisms often cluster at specific depths where oxygen, though low, remains just sufficient to sustain metabolism. The layering of life mirrors the layering of chemistry. Sediment traps deployed within the zone collect sinking particles, quantifying the flux of organic matter that drives oxygen consumption. Chemical analyses of water samples reveal intense nitrogen transformations, evidence of active microbial communities operating under limited oxygen. Together, physical, biological, and chemical measurements form a coherent portrait of a vast system shaped by persistent hypoxia. The Eastern Tropical Pacific Oxygen Minimum Zone also interacts with climate variability. El Niño and La Niña events alter surface productivity and circulation patterns, subtly shifting the intensity and vertical extent of low oxygen waters. These fluctuations ripple through ecosystems, influencing the distribution of fish, squid, and zooplankton that navigate the boundary between tolerable and limiting conditions. Along continental margins, the proximity of hypoxic waters to coastal fisheries adds complexity. Species sensitive to oxygen may alter depth ranges or migrate laterally, compressing into narrower habitats. Observations from research cruises document these shifts, linking chemical gradients to ecological response. Across this immense basin, low oxygen is neither isolated nor transient. It is woven into the fabric of the eastern tropical Pacific, maintained by the interplay of currents, productivity, and slow renewal. Within its boundaries, life arranges itself carefully, tracing invisible contours that extend across an oceanic scale. From the bright surface far above, a quiet rain begins. It is not water that falls, but fragments of life. Tiny shells from plankton, bits of algae, fecal pellets, sloughed cells, and the remains of creatures too small to notice drift downward in a steady cascade. Scientists call this continuous shower marine snow. Each particle is light, irregular, and slow, descending through hundreds of meters with almost imperceptible motion. As it sinks, the snow changes. Bacteria colonize its surface, feeding and multiplying. Small zooplankton intercept pieces along the way, nibbling and breaking larger fragments into finer ones. What finally reaches the oxygen-poor layers below is diminished, a fraction of the energy once captured from sunlight at the surface. In these midwater zones, marine snow becomes the principal source of sustenance. There are no fields of algae growing in darkness, no dense meadows of productivity. Instead, nourishment arrives sporadically, particle by particle. Organisms adapted to this environment are shaped by the rhythm of this descent. Some extend delicate appendages to intercept the falling debris. Others possess mucus nets or elongated arms that increase the likelihood of contact with drifting particles. Feeding is often passive, relying on chance encounters rather than active search. Energy gained from a single fragment may be modest, yet it can sustain a body calibrated to low demand. The slow speed of descent influences this ecology. Larger aggregates may take days or weeks to travel from surface to depth. During that time, much of their nutritional content is consumed by microbes. By the time the remnants arrive in oxygen minimum zones, they are sparse in calories. Survival depends on extracting maximum value from minimal input. Chemical composition matters as well. Marine snow carries not only carbon, but nitrogen, phosphorus, and trace elements essential for cellular processes. Even when oxygen is limited, these nutrients support growth and maintenance in animals and microbes alike. The steady, if thin, supply maintains the base of the midwater food web. Variability in surface productivity alters the volume of this snowfall. Seasonal blooms increase the flux, briefly enriching deeper layers. In quieter periods, the descent thins further, and organisms rely on stored reserves. The entire system remains tethered to events occurring far above, linked by gravity and time. In oxygen-poor waters, life does not feast. It persists on what drifts down, fragment by fragment. Marine snow binds the sunlit ocean to its dim interior, delivering sustenance across distance. Through this continual fall of organic matter, even the most constrained layers receive a trace of the surface, enough to sustain those shaped to endure on scraps. Across the surface of the ocean, a gradual warming is altering the chemistry of breath. As water temperature rises, its capacity to hold dissolved gases declines. The relationship is physical and precise. Warm molecules move more vigorously, allowing oxygen to escape more readily back into the atmosphere. Even small increases in temperature reduce the equilibrium concentration of oxygen that seawater can retain. In a warming world, the upper ocean begins with a thinner reserve. Heat also strengthens stratification. When surface layers warm, they become lighter relative to the cooler water below. The density contrast sharpens, reinforcing the thermocline that separates upper and lower layers. This intensified layering limits vertical mixing, making it more difficult for oxygen absorbed at the surface to reach deeper waters. The renewal of mid-water layers slows further. Reduced mixing carries consequences beyond simple solubility. In productive regions, organic matter continues to sink and decompose, consuming oxygen at depth. If replenishment from above weakens, the balance tilts toward depletion. Oxygen minimum zones can expand laterally and vertically, enlarging the volume of water where concentrations remain low. Observational records over the past 50 years document a measurable global decline in oceanic oxygen content. Long-term monitoring programs, combining ship-based measurements with autonomous floats and moored sensors, reveal widespread decreases across many basins. The decline is not uniform. Some regions exhibit sharper losses, particularly in the tropics and subtropics, where stratification is strong and ventilation limited. Climate variability interacts with these trends. Shifts in wind patterns, alterations in upwelling intensity, and changes in large-scale circulation influence regional oxygen levels. Yet the overarching signal remains consistent with warming-driven stratification and reduced solubility. Model projections suggest that, as temperatures continue to rise, further expansion of low oxygen zones is likely in many parts of the ocean. The implications extend through ecosystems. Species adapted to narrow oxygen ranges may find their habitable space compressed. Vertical boundaries shift upward or downward, altering migration patterns and predator-prey interactions. Microbial processes intensify in newly hypoxic regions, potentially increasing nitrogen loss through denitrification and affecting nutrient availability at the surface. In this unfolding pattern, physical and biological systems remain intertwined. Temperature alters chemistry, chemistry reshapes circulation, and circulation influences life. The ocean continues to breathe, yet its inhalations and exhalations grow uneven. Through incremental warming and reinforced stratification, the extent of thin water increases, expanding zones where oxygen becomes a limiting factor in the quiet architecture of the sea. Along continental shelves where human livelihoods meet the sea, subtle chemical shifts translate into visible change. As oxygen minimum zones expand or intensify, the vertical space available to many marine species narrows. Fish that require moderate to high oxygen concentrations avoid deeper hypoxic layers, remaining closer to the surface. When warming strengthens stratification and oxygen declines at depth, this habitable band becomes compressed between low oxygen below and, in some regions, warmer surface waters above. This vertical compression alters behavior. Pelagic species that once occupied broad depth ranges cluster within thinner strata, increasing local densities. Such crowding can intensify competition for food and heighten vulnerability to predators that share the same constrained space. The three-dimensional habitat becomes effectively two-dimensional, a narrowing corridor where movement is limited by chemistry. Commercially important fish respond in varied ways. Some shift geographically, migrating toward cooler or better ventilated waters. Along eastern boundary systems, for example, low oxygen layers can shoal toward the surface, pushing species horizontally along the coast or offshore. Fisheries that depend on predictable distributions encounter variability, as target species appear in new locations or decline in former grounds. Demersal species that dwell near the seafloor face particular challenges when hypoxic waters encroach on benthic habitats. Reduced oxygen can impair growth, reproduction, and immune function. In severe cases, mass mortalities occur when oxygen drops below critical thresholds. Even sublethal levels influence feeding rates and predator avoidance, reshaping ecological interactions. Compression also affects prey. Zooplankton and small forage fish confined to narrow bands may become more accessible to surface-oriented predators, including seabirds and marine mammals. At the same time, predators that cannot tolerate low oxygen may lose access to portions of their former hunting grounds. The balance among trophic levels shifts in response to invisible gradients. Long-term monitoring in several regions has linked declining oxygen to changes in catch composition and yield. Species tolerant of hypoxia may increase relative to more sensitive forms, altering community structure. Fisheries management must adapt to these evolving patterns, integrating chemical measurements alongside traditional stock assessments. The consequences extend beyond economics. Oxygen loss influences nutrient cycling, habitat quality, and biodiversity. Coral and sponge communities near expanding low oxygen zones may experience stress, while microbial processes intensify in newly hypoxic sediments, modifying local chemistry. In this interplay between ocean physics and human activity, the thinning of dissolved oxygen reverberates outward. Habitats contract, distributions shift, and ecological relationships adjust. The ocean continues to sustain fisheries, yet the contours of that support are reshaped by changes unfolding quietly within its layered depths. There is a boundary in the water that has no visible line, yet it is felt by every gill and membrane that crosses it. At certain concentrations, oxygen falls below the threshold required to sustain routine activity. For many marine species, this critical level lies well above zero. When dissolved oxygen declines toward these limits, physiological stress begins quietly. Ventilation rates increase as animals attempt to draw more water across their gills. Hearts beat faster, circulating what little oxygen is available to vital organs. As concentrations continue to drop, behavior shifts. Fish that once swam steadily may reduce their movements, hovering or settling to conserve energy. Some alter their depth, rising or descending in search of more favorable conditions. Others aggregate along narrow gradients, where oxygen remains marginally higher, compressing into thin layers that offer temporary refuge. Laboratory studies reveal that below species-specific thresholds, growth slows and reproduction is impaired. Enzymatic processes become constrained, and tissues receive insufficient oxygen to sustain normal function. In natural settings, these physiological pressures translate into redistribution. Sensitive species gradually withdraw from hypoxic zones, abandoning habitats that no longer meet their metabolic needs. The retreat is not uniform. Some organisms tolerate lower levels through specialized adaptations in blood chemistry, gill structure, or metabolic regulation, yet even among these, limits exist. When oxygen approaches near zero, only a small subset of specialists remains, individuals whose physiology has evolved to function at the margins of aerobic life. This narrowing of community composition reshapes ecological interactions. As generalist species depart, the assemblage becomes dominated by tolerant forms, often smaller in size and slower in activity. Food webs simplify further, reflecting the constrained energy landscape. The absence of certain predators or competitors can create temporary shifts in abundance among those that stay. At the extreme edge, microbial processes intensify. Bacteria capable of anaerobic metabolism expand their influence, further altering chemical conditions. The water becomes a zone defined less by active swimming and more by chemical exchange and minimal motion. Field measurements document these gradients with increasing precision. Oxygen sensors lowered through the water column trace steep declines over short vertical distances, marking the transition from tolerable to limiting conditions. Acoustic surveys reveal corresponding changes in biomass distribution, with dense layers thinning abruptly below certain depths. Within this invisible boundary, survival depends on alignment between physiology and environment. As oxygen wanes, many species step back, seeking more breathable waters elsewhere. Those that remain embody the outer limits of aerobic endurance, inhabiting a realm where the margin between persistence and absence is measured in fractions of a milliliter per liter. In water, where oxygen is scarce and motion is measured, light becomes a language of restraint. Many midwater organisms produce bioluminescence, generating light through controlled chemical reactions within specialized cells. The process typically involves luciferin, a light-emitting molecule, and luciferase, the enzyme that catalyzes its oxidation. Even in low oxygen environments, only small amounts are required for these reactions, making light production energetically efficient when used sparingly. In thin waters, bioluminescent displays are rarely prolonged. Short flashes replace sustained glows. A brief pulse may startle a nearby predator, illuminate a drifting particle, or signal the presence of a mate. Because rapid swimming is costly in hypoxic layers, communication that relies on light rather than speed offers advantage. A flicker can travel outward without the sender expending significant oxygen in pursuit or escape. Some species emit controlled bursts from photophores arranged along their bodies. These light organs are often shielded and directed, focusing emission downward or laterally. The intensity and duration are finely tuned. A pulse lasting less than a second can convey information while conserving metabolic reserves. The reaction ceases as quickly as it begins, leaving darkness intact. Counter-illumination provides another strategy. In regions where faint downwelling light penetrates from above, certain fish and squid produce a soft glow along their ventral surfaces. By matching the intensity of light filtering downward, they reduce their silhouettes when viewed from below. This camouflage through light allows them to remain concealed without rapid movement. Adjustments in brightness are achieved through neural control of photophores, responding to subtle changes in ambient illumination. In oxygen minimum zones, where vertical compression concentrates organisms into narrower bands, such visual strategies become particularly useful. Encounters occur within constrained space, and the ability to signal or obscure without extensive locomotion preserves energy. Light travels efficiently in water, extending the reach of communication beyond what a slow body might accomplish through motion alone. The chemistry of bioluminescence is integrated with broader metabolic regulation. Because oxygen is involved in the light-producing reaction, organisms must balance its use carefully. The small quantities required make brief flashes feasible even under limited supply. The result is a pattern of intermittent illumination, scattered points appearing and vanishing in darkness. Within these subdued layers, bioluminescence does not blaze continuously. It flickers with intention, economical, and precise. Light becomes a tool adapted to scarcity, enabling interaction and concealment while respecting the limits imposed by thin water. At first glance, the inhabitants of these layers seem fashioned from another imagination. Bodies appear translucent, edges softened, forms elongated or compressed in ways unfamiliar to surface eyes, yet their shapes are not whimsical. They are precise responses to darkness and limited oxygen, each feature shaped by physics and chemistry rather than spectacle. Transparency offers concealment without effort. In dim mid-water, where faint residual light filters downward, a solid silhouette can betray presence. Tissue composed largely of water bends and transmits light rather than blocking it, reducing visibility from multiple angles. Because gelatinous material contains little dense muscle or bone, it also requires less oxygen to maintain. A nearly transparent body becomes both camouflage and metabolic economy combined. Where skeletons do exist, they are often reduced. Bones may be thinner, less calcified, or partially replaced by cartilage. Heavy structural elements demand resources to build and maintain, and dense muscle anchored to rigid frames consumes oxygen during activity. In thin water, buoyancy can be achieved through lipids or specialized tissues that match the density of surrounding seawater. The need for robust skeletal support diminishes when rapid acceleration is rare. Sensory organs, by contrast, may be enlarged. Eyes grow wide to capture scarce photons. Tubular or upward-oriented structures concentrate faint light from above. In other species, vision yields priority to mechanoreception, with elongated fins or whisker-like appendages detecting subtle vibrations. Chemoreceptors become finely tuned, sensing minute gradients in dissolved compounds. Enlarged sensory surfaces allow organisms to perceive without constant movement, conserving oxygen by replacing search with detection. Internal organs reflect similar refinement. Gills may present increased surface area relative to body size, while circulatory systems emphasize efficiency over speed. Mitochondria within cells operate steadily at low flux, extracting energy from minimal supply. Even pigmentation adjusts. Dark hues absorb residual light and reduce reflectance, aiding concealment in a dim environment. Pressure at depth further shapes form. Flexible bodies accommodate compression without rigid resistance. Gas-filled cavities are minimized or absent, preventing collapse. The result is an architecture that seems delicate yet endures immense hydrostatic force. To surface observers, such creatures appear alien because they diverge from familiar proportions. Limbs elongate, heads become transparent domes, tissues shimmer faintly. Yet each deviation traces back to constraint. Darkness favors sensitivity over ornament. Low oxygen rewards thrift over mass. Buoyancy replaces bulk, and perception substitutes for pursuit. In this layered ocean, strangeness is simply adaptation rendered visible. What seems otherworldly is, in truth, an elegant alignment with conditions few terrestrial beings encounter. The forms drifting in thin water embody the quiet logic of survival, shaped by shadow and scarcity. Beneath the movements of drifting bodies and the slow pulse of circulation, another story unfolds in molecules. When oxygen dwindles, the chemistry of seawater shifts in subtle yet measurable ways. Microbial communities adjust their metabolic pathways, and in doing so, they leave behind distinct chemical traces. One of these is nitrous oxide, a gas produced during the incomplete reduction of nitrate in low oxygen conditions. In waters where oxygen is scarce but not entirely absent, denitrifying bacteria generate nitrous oxide as an intermediate. Some of it escapes further reduction and accumulates, later diffusing toward the atmosphere. Concentrations of nitrous oxide often peak at the upper boundaries of oxygen minimum zones, forming a thin layer enriched relative to surrounding waters. This enrichment serves as a chemical fingerprint, signaling zones where aerobic respiration gives way to alternative pathways. Measurements of dissolved gases reveal these gradients with precision, mapping invisible transitions through shifts in concentration alone. Carbon cycling also transforms under hypoxic conditions. In oxygen-rich environments, organic carbon is oxidized efficiently to carbon dioxide. As oxygen declines, the sequence of electron acceptors used by microbes changes, altering the rates and byproducts of decomposition. Some carbon is processed more slowly, extending its residence time in the water column. In sediments beneath low oxygen waters, organic matter can accumulate more readily, preserved by limited aerobic breakdown. The balance between dissolved inorganic carbon and alkalinity shifts subtly in response to these processes. Respiration increases carbon dioxide concentrations, influencing local pH and buffering capacity. Although these changes may be modest on short timescales, over broader regions they reflect the integrated activity of microbial communities operating under constraint. Trace metals and reduced compounds can also serve as indicators. Manganese and iron may change oxidation state when oxygen is limited, becoming more soluble and mobile. The presence of hydrogen sulfide in extreme cases signals a transition from hypoxia to anoxia, though such conditions are rare in open ocean oxygen minimum zones. Each chemical species responds predictably to the availability of oxygen, forming a layered pattern of reactions. Scientists track these transformations through water sampling and sensor technology capable of detecting minute variations. Profiles of dissolved gases, nutrients, and redox-sensitive elements together outline the chemistry of a breathless sea. The gradients are not chaotic but structured, reflecting the interplay of circulation, biological demand, and limited supply. In these waters, chemistry becomes both consequence and cause. The decline of oxygen initiates alternative metabolic routes, and the products of those routes influence broader cycles of nitrogen and carbon. Invisible yet pervasive, these chemical signatures mark the presence of hypoxia, recording in dissolved form the adjustments of life to scarcity. Long after research vessels depart and the surface grows still, instruments continue their quiet patrol. Autonomous floats drift through the ocean, rising and sinking on programmed cycles that span thousands of kilometers and many years. Carried by currents, they adjust buoyancy with small internal pumps, descending into dim layers and returning to the surface at regular intervals. During each ascent, sensors record temperature, salinity, and, increasingly, dissolved oxygen with remarkable precision. Modern oxygen sensors rely on optical principles rather than fragile chemical titrations. Many use luminescent films whose glow changes subtly in the presence of oxygen. As dissolved oxygen interacts with the sensing surface, it quenches the emitted light in measurable ways. By tracking these changes, floats calculate concentrations with accuracy sufficient to detect long-term trends. Once at the surface, the data are transmitted by satellite, joining global archives in near real time. Across ocean basins, networks of these profiling floats form a distributed observatory. They reveal the vertical structure of oxygen minimum zones, capturing seasonal shifts and gradual expansion. Because floats operate continuously, they document changes that brief expeditions might miss. Patterns emerge from repeated profiles, outlining gradients that fluctuate with climate variability and circulation. Deep-sea robotics extend this perspective. Remotely operated vehicles and autonomous underwater vehicles travel horizontally through low oxygen layers, carrying high-resolution sensors and cameras. They can hover within narrow chemical boundaries, measuring oxygen alongside nitrate, pH, and other tracers. Some platforms deploy microelectrodes capable of detecting fine-scale variations over centimeters, illuminating structures within the broader zone. These technologies allow scientists to map oxygen not as a static field, but as a dynamic system. Eddies, internal waves, and mesoscale currents distort the boundaries of hypoxia, creating pockets where concentrations rise or fall temporarily. Real-time measurements capture these transient features, linking physical motion to chemical response. In addition to floats and vehicles, moored arrays anchored to the seafloor provide continuous time series at fixed locations. Combined, these tools generate a layered view of the ocean's breathing, from basin-scale circulation down to localized fluctuations. The integration of data from autonomous platforms has refined understanding of how oxygen minimum zones evolve. Subtle declines over decades become visible when profiles are compared across time. Expansions and contractions can be tracked with spatial clarity previously unattainable. In the quiet depths where life adapts to thin water, machines now drift and glide, measuring without disturbance. Through their persistent presence, the contours of oxygen are traced in detail, revealing a living ocean whose chemistry shifts in patterns both gradual and complex. The ocean does not inhale and exhale in hours or days. Its breathing unfolds across centuries. At high latitudes, where winter air chills the sea surface, water grows dense and heavy. As it cools and in some regions becomes saltier through ice formation, it sinks, descending into the abyss. This process initiates the thermohaline circulation, a vast conveyor driven by differences in temperature and salinity. From polar basins, deep currents spread outward, slipping along the seafloor and weaving between continents. These deep waters carry with them the oxygen absorbed at the surface. When they first sink, they are rich in dissolved gas, charged with the imprint of cold air. Yet as they travel through the interior of the ocean, that reserve is gradually consumed by organisms and microbes. Respiration continues in darkness, drawing down oxygen molecule by molecule. The journey is slow. Water that sinks in the North Atlantic may take hundreds to more than 1,000 years to return to the surface in distant regions. Along the way, it mixes subtly with surrounding masses, exchanging heat and dissolved substances. By the time it rises again through upwelling zones, its oxygen content is diminished, its chemical composition altered by long residence in the deep. This circulation connects basins into a single planetary system. The Pacific, Indian, and Atlantic Oceans are linked by pathways that redistribute heat, carbon, and nutrients. Oxygen minimum zones are embedded within this larger framework, shaped in part by how recently their waters were last in contact with the atmosphere. Regions ventilated frequently tend to maintain higher oxygen, while those isolated for centuries bear the mark of prolonged consumption. Climate variability can influence the strength and pattern of thermohaline circulation. Changes in temperature, freshwater input from melting ice, or wind-driven currents alter density gradients that govern sinking and spreading. Because the system responds over long timescales, its adjustments are gradual yet profound. Seen in this light, the ocean's respiration resembles a slow planetary pulse. Surface exchange with the atmosphere represents inhalation, while the sinking of dense water distributes that breath inward. Deep renewal through upwelling completes the cycle, returning altered water to sunlight, where it can again absorb oxygen. Within this expansive rhythm, low oxygen zones reflect the balance between supply delivered by circulation and demand generated by life. The timing of renewal, measured in centuries, sets the background against which shorter-term fluctuations play out. The sea breathes steadily, its cadence shaped not by minutes but by generations of water moving through the dark. Looking forward through the lens of climate models, the contours of low oxygen water extend beyond their present boundaries. Earth system simulations that integrate ocean circulation, atmospheric warming, and biogeochemical cycles project continued declines in dissolved oxygen through the year 2100. As surface temperatures rise and stratification strengthens, the supply of oxygen to intermediate depths is expected to weaken in many basins. Even modest percentage losses translate into substantial changes across vast volumes of water. Model projections indicate that tropical and subtropical regions are particularly vulnerable. In these areas, oxygen minimum zones already exist near critical thresholds. Further warming is likely to expand their vertical and horizontal extent, compressing habitable space for species sensitive to low oxygen. The eastern tropical Pacific and parts of the Arabian Sea, long-established centers of hypoxia, may intensify under continued greenhouse gas emissions. High-latitude regions are not exempt. Changes in sea ice cover and freshwater input can alter the formation of dense waters that ventilate the deep ocean. If deep water production slows or shifts location, renewal of oxygen at depth may diminish over time. Because thermohaline circulation operates on long timescales, adjustments may unfold gradually yet persistently. Quantitative estimates vary among models, reflecting differences in assumptions about emissions, scenarios, and climate sensitivity. Under high-emission pathways, global ocean oxygen inventories are projected to decline by several percent by the end of the century. Regional changes may be larger, with some mid-water layers experiencing more pronounced reductions. These projections are grounded in observed trends over recent decades. Measurements already document widespread deoxygenation in many parts of the ocean. Models that successfully reproduce past changes lend confidence to their forward simulations, though uncertainty remains regarding the magnitude and spatial pattern of future decline. Regions with limited ventilation and high biological productivity appear most at risk. Coastal upwelling systems, semi-enclosed seas, and areas of strong stratification may see hypoxic layers shoal closer to the surface. Such shifts would influence ecosystems and fisheries as species distributions adjust to narrowing oxygen windows. At the same time, natural variability will continue to modulate regional patterns. Climate oscillations, changes in wind fields, and shifts in ocean currents will overlay shorter-term fluctuations upon long-term trends. The projected expansion of low oxygen zones does not imply uniform change, but rather a complex mosaic of intensifying and shifting boundaries. Within these modeled futures, the ocean's breath becomes more uneven. The balance between oxygen supply and biological demand tilts subtly yet steadily in many regions. By 2100, the spatial footprint of thin water is likely to be larger than it is today, reflecting the intertwined influence of warming, circulation, and life itself. If oxygen continues to decline beyond present levels, the rearrangement of life will not occur all at once, but through gradual shifts in space and balance. As hypoxic layers expand upward or outward, species that require higher concentrations will adjust their ranges. Some will migrate horizontally, tracking cooler or better ventilated waters along coastlines or toward higher latitudes. Others will move vertically, compressing into thinner surface bands where oxygen remains sufficient. These redistributions may unfold over seasons or decades, guided by physiological limits rather than sudden collapse. Migration, however, is not equally available to all. Sessile organisms and species tied to specific habitats, such as certain benthic invertebrates, cannot relocate easily. When low oxygen intrudes into their environment, survival depends on tolerance rather than movement. If thresholds are exceeded, local populations may decline or disappear, leaving gaps in the ecological network. As species shift or withdraw, food webs reorganize. Predators may lose access to customary prey if distributions no longer overlap. Prey populations may expand temporarily in the absence of certain hunters, only to encounter new pressures as different predators move in. The structure of interactions becomes fluid, reflecting the altered geometry of habitable space. In some regions, the shoaling of hypoxic waters could intersect with warming surface temperatures, creating narrow windows where both oxygen and thermal conditions are tolerable. Such double compression intensifies competition within constrained layers. Behavioral changes, altered growth rates, and shifts in reproductive timing may follow as organisms adapt to the new configuration. The possibility of tipping points arises when incremental change leads to abrupt ecosystem transitions. For example, sustained oxygen loss in coastal systems can favor microbial processes that release additional nutrients or greenhouse gases, reinforcing local chemical shifts. If feedbacks strengthen, recovery to prior states may become difficult even if external conditions stabilize. Deep ocean processes also respond. Expanded low oxygen zones can increase denitrification, reducing the availability of fixed nitrogen for surface productivity. Changes in nutrient balance may ripple upward, influencing primary production and, in turn, the export of organic matter that drives oxygen consumption at depth. The system becomes interconnected across scales. Model studies suggest that while global extinction is unlikely under projected scenarios, regional biodiversity patterns may alter significantly. Species with narrow oxygen tolerances face greater risk, whereas those capable of metabolic flexibility may gain relative prominence. Over time, community composition could diverge from historical baselines. If oxygen falls further, the ocean will not become empty, it will reorganize. Migration, reshuffling, and threshold responses will shape the emerging landscape. The transformation will reflect the same principle that governs thin water today, that life persists within limits, adjusting its distribution and relationships to the chemistry that surrounds it. In the quiet persistence of these waters, survival has taken on a different meaning. Here, endurance does not belong to the swiftest swimmer or the most aggressive hunter. It belongs to those who spend little and waste nothing. Across generations, natural selection has favored efficiency over speed, refining bodies and behaviors that conserve energy at every level. Metabolic rates among many inhabitants are markedly lower than those of relatives in more oxygen-rich environments. Cells operate at reduced demand, enzymes tuned to function effectively even when oxygen concentrations are thin. Rather than sustaining high activity, these organisms maintain a careful balance between intake and expenditure. Growth may be slower, reproduction less frequent, yet persistence remains steady. Energy thriftiness appears not only in physiology, but also in form. Gelatinous tissues, sparse musculature, and minimal skeletal investment reduce the cost of maintenance. Some species exhibit reduced visual systems or streamlined organs, allocating resources only to functions essential for survival in dim, low-oxygen layers. Each anatomical feature reflects a long history of selection under constraint. Behavior follows the same principle. Movement is measured, often limited to brief adjustments rather than sustained pursuit. Feeding strategies rely on interception, filtration, or opportunistic capture rather than active chase. By aligning behavior with environmental limits, these organisms avoid the metabolic spikes that could prove fatal in thin water. At the biochemical scale, adaptations such as high affinity, respiratory pigments, and efficient oxygen extraction from surrounding water illustrate fine-tuned optimization. Gills may be enlarged relative to body size, circulatory systems structured to maximize transfer with minimal energy cost. Even heart rates and ventilation patterns can adjust to conserve reserves when external concentrations decline. Evolution in these zones has not produced extravagance but restraint. Traits that demand constant high oxygen supply are gradually filtered out, while those compatible with scarcity persist. Over time, this selective pressure shapes communities composed of specialists, each adapted to narrow but stable conditions. These lessons extend beyond individual species. They reveal how life can reorganize priorities under limitation. Instead of competing for dominance through speed or size, success emerges from patience, moderation, and alignment with available resources. Within the broader ocean, such strategies may seem subdued, yet in regions where oxygen thins, they represent a quiet mastery. Survival becomes an exercise in balance, an intricate calibration between need and supply. The inhabitants of these zones demonstrate that resilience often resides not in intensity, but in careful stewardship of energy. In their slow rhythms and economical designs, the survivors embody an evolutionary response to constraint, showing that even in diminished breath, life can refine itself toward efficiency and persistence. For a long time, these waters were described with a single word that implied absence. Charts labeled them as dead zones, a phrase that suggested vacancy and silence. Early sampling methods, limited by coarse nets and brief expeditions, often returned sparse catches. From those fragments grew the impression of emptiness, as though low oxygen erased life altogether. Yet the view was incomplete. As instruments improved and observations grew more patient, the outline of a different reality emerged. Cameras revealed drifting invertebrates suspended in delicate balance. Acoustic surveys detected layers of organisms arranged along precise chemical boundaries. Microscopic analyses uncovered dense microbial communities orchestrating complex cycles beyond the reach of earlier tools. The misconception rested not only on technology but on expectation. Life in thin water does not resemble the bustling reefs or teeming coastal shelves that often define marine abundance. It is subtler, organized around limitation rather than surplus. Density may cluster within narrow bands where oxygen and nutrients align just enough to permit survival. In some regions, biomass accumulates at the margins of hypoxic layers, forming concentrated assemblages. These aggregations can rival or exceed those in more oxygenated depths, though composed of different species and structures. Gelatinous forms, small crustaceans, specialized fish, and vast populations of bacteria contribute to a hidden abundance that conventional nets once overlooked. Even within the lowest oxygen cores, life persists at microscopic scales. Anaerobic processes, alternative metabolisms, and symbiotic relationships sustain communities adapted to scarcity. What appears barren to one method of observation may, under another lens, reveal intricate activity. Thriving in this context does not imply rapid growth or dramatic display. It means maintaining function within constraint, reproducing at sustainable rates, and sustaining ecological interactions over time. The metrics of success shift. Instead of high diversity of large, mobile species, there may be dominance by fewer, highly specialized organisms. Instead of swift predation, there may be slow filtration and careful energy exchange. As understanding deepens, the term dead zone becomes a simplification rather than a literal description. These areas are defined by reduced oxygen, not by absence of life. They host communities shaped by chemistry, circulation, and evolutionary history. Seen through this broader perspective, thin waters are not voids but alternative realms. Their inhabitants operate under different rules, yet they contribute to the ocean's overall functioning. Nutrient cycling, carbon processing, and trophic interactions continue, though arranged in patterns distinct from oxygen-rich systems. The recognition of hidden abundance alters the narrative. What was once imagined as empty space becomes a landscape of adaptation and persistence, quieter perhaps, but alive in forms attuned to its limits. Long before instruments traced gradients and models projected futures, the ocean had already been breathing in its own slow rhythm. Across geological time, oxygen concentrations have risen and fallen in response to shifts in climate, circulation, and life itself. During certain ancient intervals, widespread anoxia expanded through large portions of the ocean, leaving dark layers in marine sediments as testimony. At other times, vigorous circulation renewed deep waters more efficiently, increasing oxygen supply to remote basins. These oscillations unfolded over millions of years, guided by tectonic rearrangements, volcanic emissions, and evolutionary innovation. The burial of organic carbon in sediments gradually enriched the atmosphere with oxygen, while the weathering of continents altered nutrient delivery to the sea. Each process influenced marine chemistry, creating feedbacks between land, ocean, and air. The deep sea, vast and interconnected, acted as a reservoir where these changes were moderated and stored. Thermohaline circulation, driven by density differences, continued its patient turnover. Even as surface conditions fluctuated, deep currents redistributed heat and dissolved gases, smoothing extremes across space and time. In this way, the ocean functioned as a quiet regulator of planetary balance, absorbing carbon, buffering temperature, and exchanging oxygen with the atmosphere. Life persisted through these transitions. Species diversified, adapted, and sometimes vanished, yet ecosystems reassembled in new configurations. Periods of low oxygen did not eliminate life from the sea, they reshaped it. Microbial communities adjusted metabolic pathways, multicellular organisms evolved tolerances, and new niches emerged within altered chemistry. Today's oxygen minimum zones exist within this longer narrative. Their boundaries may expand or contract under contemporary climate change, but they remain part of a system that has fluctuated before. The pace of current change is rapid relative to geological timescales, yet the fundamental processes of circulation and biological adaptation continue to operate. Beneath the surface, renewal proceeds slowly. Dense waters still sink at high latitudes, carrying dissolved oxygen downward. Upwelling regions still return deep water to sunlight, where it can exchange gases once more. Microbes, plankton, invertebrates, and fish continue their interactions within the limits set by chemistry and physics. The ocean's breathing is not dramatic in the manner of storms or breaking waves. It is measured in gradients, currents, and cycles that span generations of water. Oxygen levels rise and fall, shaped by forces both natural and human, yet the underlying rhythm endures. In the deep sea's darkness, survival remains steady. Adaptation, circulation, and chemical exchange weave together in a continuous process. The ocean continues to breathe, not in haste, but with a persistence that has guided life through ages of change.